Electrophoresis of DNA restriction fragments allows separation based on size to be carried out. However, it provides no indication as to the presence of a specific desired fragment among the complex sample. This can be achieved by transferring the DNA from the intact gel onto a piece of nitrocellulose or nylon membrane placed in contact with it. First, the gel is soaked in alkali to render the DNA single-stranded. It is then transferred to the membrane so that the DNA becomes bound to it in exactly the same pattern as that was originally on the gel. And this is called southern blot. The point of this operation is that the membrane can now be treated with a labeled DNA molecule, which we called it a gene probe. And a series of washing steps with buffer is then carried out to remove any unbound probe, and the membrane is then developed, after which the precise location of the probe and its target may be visualized. Sometimes it is also possible to analyze DNA from different species of organism by blotting the DNA and then using a gene probe representing a protein or enzyme from one or the other organism. In this way, it is possible to search for related genes in different species. And this technique is a little bit modification of the southern blot, and we call it zoo blotting. While very similarly, the same basic process of nucleic acid blotting can be used to transfer RNA from gel onto similar membrane. This allows the identification of a specific mRNA sequences of a defined length by hybridization to a labeled gene probe, and it's known as northern blotting or northern blot analysis. It is possible with this technique to not only detect specific mRNA molecules, but it may also be used to quantify the relative amount of specific mRNA species in the sample. It is useful to separate the mRNA transcript by gel electrophoresis under denaturing conditions, since this improves the resolution and allows a more accurate estimation of the size of the transcripts. The format of the blotting may be altered from transfer from a gel to a different application to slot on a specific blotting apparatus containing the nylon membrane. This is termed as dot blotting. However, it does not specifically provide the information regarding the size of the fragments.